DNA chips. So what are DNA chips? Well, DNA chips are used to study which genes are active and inactive in human beings. They help scientists to understand how and why some genes don't function properly, and help greaten the amount of genes scientists can look at once. So how do DNA chips work? DNA chips are made by a robotic machine that arranges very small amounts of thousands of genes onto a single microscopic slide. When a gene is activated, cellular machinery will start to copy certain parts of that gene. This creates mRNA. This mRNA is complementary and will bond to the DNA from which it originated. In order to determine which genes are activated, a scientist has to collect the mRNA in the cell. Then the researcher labels each mRNA with a reverse transcriptase enzyme, which creates cDNA that is complementary to the mRNA. So if you look here on this diagram, you extract the mRNA from whatever group of cells you have, whether it's cancer cells or normal cells, and so this is the mRNA, and it gets reverse transcripted into cDNA, so those are able to bond. And then fluorescent nucleotides are attached to the cDNA. So then when the researcher puts the labeled cDNA onto a DNA microarray slide, a special scanner measures the fluorescent intensity of each area of the slide. So when a gene is active, it produces many mRNA molecules and makes a very fluorescent and bright area, and if a gene is inactive, they produce fewer mRNA and therefore appear less fluorescent. So this is how the researchers can tell if the gene is activated or not. So what can DNA chips be used for? Well, microarray technology helps researchers to learn about heart disease, mental illnesses, infectious diseases, and many other diseases. Researchers also use microarray to further classify cancers by seeing which genes are present in different tumors based on the gene activity. This greatly helps in making more personalized treatment for each patient because they know what kind of genes they're dealing with. Scientists will also know how treatments affect tumors by looking at how present the gene is in the tumor versus a normal group of cells after treatment.